Today we're going to discuss ion channels and neuronal communication. This is a general overview of the topics that we'll discuss today. As you can see, there are many components involved with neuronal communication, ion channels being one of those major components. We'll discuss the communication between neurons in depth and how ion channels assist in that communication. Now, what is neuronal communication? It's the function of a neuron. The function of a neuron is to receive input information from other neurons, process that information, and then send the information as output to other neurons. So, neuronal communication is basically neurotransmission to neurons communicating between themselves. Neurons process all the information coming in and out of the central nervous system, which is a vital part of the body, without which we would not be able to function as human beings. Now, neurotransmission is accomplished by the movement of chemicals or electrical signals across a synapse, and we'll discuss a synapse in the next slide. So let's take a look at the neuron. Take a look at the picture on the right. This gives a general picture of a neuron. As you can see, there's little tendrils coming off either end, and this is how they connect through the synaptic space and communicate with each other. Those projections will get close to each other and exchange chemicals and signals and all of that. So a neuron is a specialized cell that transmits chemical and electrical signals in the brain. And they are the basic building blocks of the central nervous system. As I mentioned before, the central nervous system is very important to the overall function of the human body. So therefore, neurons are very important as well. Now, there's 4 primary components of a neuron. There's the soma, which is the cell body, the axon, which is a projection that conducts electrical impulses away from the soma. So you can see the axon is one of those where the neuron starts to move away from the body. There's dendrites, which are projections coming off of the axon. They receive messages from other neurons. And then the synapse is a junction between neurons. So those are important components of the axon, or the neuron, that you'll need to know. There are other components of the neuron that we won't discuss today, as they're not super important to neuronal communication. However, they are important to neuronal function. Now, each neuron has a charged cellular membrane. And the charge of this membrane can change in response to a neurotransmitter molecule that's released from other neurons and stimuli. And we're going to watch this short video that kind of describes the structure of neurons in depth.
This is a brain. Estimates vary, but right now the best guess seems to be that our brains contain around 85 billion neurons. The neuron is a nerve cell, and it's the primary functional unit of the nervous system. This is a generic image of a neuron. Neurons actually come in all shapes and sizes, but this is the prototypical version of a neuron that you'll often see in a textbook. The structures extending from the left side of the neuron that look a little bit like tree branches are called dendrites. Dendrites are the area where neurons receive most of their information. There are receptors on dendrites that are designed to pick up signals from other neurons that come in the form of chemicals called neurotransmitters. Those signals picked up by dendrites cause electrical changes in a neuron that are interpreted in an area called the soma, or the cell body. The soma contains the nucleus, which contains the DNA, or genetic material of the cell. The soma takes all the information from the dendrites and puts it together in an area called the axon hillock. If the signal coming from the dendrites is strong enough, then a signal is sent to the next part of the neuron, which is called the axon. At this point, the signal is called an action potential. The action potential travels down the axon, which is covered with myelin, an insulatory material that helps to prevent the signal from degrading. The last step for the action potential is the axon terminals, also known as synaptic buttons. When the signal reaches the axon terminals, it can cause the release of neurotransmitter. These purple structures represent the dendrites of another neuron. When a neurotransmitter is released from axon terminals, it interacts with the receptors on the dendrites of the next neuron, and then the process repeats with the next neuron.
So as you can see, the structure of the neuron is very important because if there's anything wrong with the neuron itself, then the communication between neurons will be inhibited or altered. And the most important concept that we'll focus on today is action potential and the ion channel's role in the action potential. So what's an ion channel? An ion channel is a pore-forming protein that allows the flow of ions across membranes. To enter or exit the neuron, ions must pass through ion channels that span the membrane. Now there's 3 different configurations of ion channels. There's open, closed, and inactive. There are multiple types of regulation of these channels. There's ligand-gated, which opens due to a ligand binding to the extracellular region of the cell. There's mechanically gated, which opens due to a physical distortion of the cell membrane. There's a leakage channel, which is randomly gated. And then the one that we're going to focus on today the most is the voltage-gated channel, which responds to changes in the electrical properties of the membrane. The key word there is electrical properties. When we're talking about action potentials today, we are discussing electrical signals being sent. So it's very important to understand that. So voltage-gated ion channels, they are often selectively permeable, which means they only let specific ions in and out of the cell. They won't just allow anyone into the cell. That could alter communication pathways, could cause a lot of problems, so they're selectively permeable. Permeable just means to pass through, so selectively they choose what can pass through the membrane. They change their structure in response to voltage changes. So voltage is a key concept here. When we're talking about action potential, we're basically talking about a change in voltage. So the structure of these voltage-gated ion channels will be changed when the voltage changes. And that's important for the types of ions that it lets through. And again, it is selectively permeable, so it does make that decision based on its structure. They regulate the relative concentrations of different ions inside and outside of the cell. So they receive signals to let them know, hey, there's too much sodium inside the cell or outside the cell. We need to move some through so that we can, balance it out, homeostasis. So let's talk about membrane potential. We discussed earlier that there is a voltage within the membrane of each neuron. Now the resting membrane potential is the steady state of the cell. This is homeostasis that we're talking about, regulated by leakage channels and these and active transport pump channels. So the leakage and the ion pumping, they receive signals to let them know there's too much of one ion inside or outside of the cell, and they regulate that passively. The resting membrane potential, it won't change without outside influence. So usually an electrical or chemical change needs to happen in order to change the resting membrane potential. The voltage at resting membrane potential is negative 70 millivolts. It's very important to remember. That's going to be our starting point for action potential. So what is action potential? Action potential occurs when an electrical signal disrupts the resting membrane potential and the balance of sodium and potassium within a cell membrane. So those are our 2 most important ions, sodium and potassium. It requires passive transport and active transport pumps to generate a transmembrane potential and an action potential. So the impulse travels along the axon via depolarized voltage-gated ion channels in the membrane. So once the signal leaves the soma, hits the axon, it's going to travel down the neuron and depolarize as it goes. Now, there's 3 different stages of action potential. There's depolarization, repolarization, and the refractory phase. Some will say there's a 4th stage called hyperpolarization. We're going to use that kind of as a little landmark rather than a separate stage. And we'll talk about that here in a second. Depolarization is caused by a couple different mechanisms. There's ligand-gated sodium channels, which open when a neurotransmitter binds to it, so that's more of a chemical reaction. There's mechanically gated sodium channels, which will open when a physical stimulus affects a sensory receptor. And then there's those voltage-gated sodium channels that we're talking about, which opens after the membrane reaches negative 55 millivolts. Now, this is called threshold when the membrane is at negative 55 millivolts. There's 2 different types, 2 different phases really of the voltage-gated sodium channel. The activation gate opens when the membrane potential crosses the threshold at negative 55 millivolts. And then there's the inactivation gate, which closes after a period of time. Now, it's important to note that depolarization happens rather quickly. The sodium channels do open very quick and they close very quick. So sodium starts to rush into the cell and the membrane becomes less negative as more sodium goes into the cell. During repolarization, it's dependent on voltage-gated potassium channels. So if you're looking at the chart, you see that spike. The left side of the spike is sodium, the right side is potassium. Now these channels open at negative 50 millivolts, so the sodium has caused the cell to become less negative at negative 50 millivolts, and so that causes the potassium gates to open, and they open slower than the sodium gates. And they open just as the voltage-gated sodium channels are inactivated. So we talked about the activation gate and the inactivation gate. Right as the inactivation gate closes, the potassium gate opens, and it also closes slowly. So it opens slowly, closes slowly. This allows a lot of potassium to get into the cell over time rather than just in a short period of time. To leave the cell. And potassium continues to leave the cell for a little while, and the membrane potential becomes even more negative as it goes past the threshold. In the negative direction, and this is called hyperpolarization. If you look back at the chart, you can see the dip below the negative 70, and that's characterized as hyperpolarization. Now, the refractory phase is the last phase of the action potential. This is basically the cell going back to homeostasis. So while an action potential is taking place, another one can't start. They're not allowed to happen at the same time in one cell just because of the amount of ions rushing in and out. There's not enough time for the cell to regulate itself if the action potentials are constantly firing within one cell. That doesn't mean that they don't happen rather quickly, that this whole process actually is just a couple milliseconds long, as you can assume, since it is our brain, functioning and these neurons communicating within the brain. That does have to happen very quickly, but it just can't happen all at once in one cell. Now, there's 2 phases of the refractory phase. There's the absolute refractory phase, which the inactivation gate of the voltage-gated sodium channel is closed at that time. So right as the inactivation gate closes, that's the absolute refractory phase. The relative refractory phase begins when the potassium gate closes. So the cell is already pretty much back at its homeostatic state. However, there's still some potassium leaking out. So once the potassium gate closes, then the cell can begin a new action potential when the signal is received. These are the resources that we use today if you want to look into any more journal articles related to neuronal communication, ion channels, and action potential.